Good evening, students. So in yesterday's class we have learned that what is cell, how it is been discovered by the scientist Robert Hooke, what are the different types of organisms based on the number of cells present in their body, what is the living organism made up of, and what is the structural organization of any cell present in our body. So while discussing the structural organization of cell in our body, we have come to know that every cell is made up of three basic components or parts. The first one was plasma membrane, which is the other name of cell membrane. The second part was cytoplasm, and the third part was nucleus. We have also seen that in addition to these three components, plant cells have another outermost covering, rigid covering, which is known as cell wall. Then we have discussed about the plasma membrane, what are its compositions, how it helps, and we have seen that how the movement of molecules take place through the plasma membrane. Okay, in that we have seen that there are two basic processes such as osmosis and diffusion. Using this process, movement of molecules takes place across the plasma membrane of any cell. Okay, so today we will see that there are three types of solutions: hypotonic solution, isotonic solution, and hypertonic solution. Okay, so in the previous video which I have sent you, in that it is briefly explained what is hypotonic solution, what is isotonic solution, and what is hypertonic solution. And it has also been shown in the video that what happens in these three types of solution when a cell is placed in this type of solutions separately. Okay, so here are the slides which describes you about the basics of these three types of solution. So you know hypotonic solution, hypo means less, so tonic means solutes. So if in the solution the amount of the salts or sugar present is very less, then that type of solution is known as hypotonic solution. Okay, so we can say that a solution is mostly made up of solute and solvent. Generally water is considered as the universal solvent. So water, and it is a mixture of solutes. So in hypotonic solution, solute concentration is very low. That means the water concentration is very high. That means the solution is dilute. So if the medium surrounding a cell has higher water concentration than the cell, that means the outside the cell the concentration is very dilute. Okay, that means inside the cell the concentration of water is less. As a result, what will happen? The water from outside medium will enter into the cell. Okay, and as a result the cell will swell up. So this is one example. So for showing the example, I have taken in the slide red blood cells, RBC. Okay, so you have kept red blood cell in a solution that is hypotonic. It means outside the water concentration is very high and inside the cell the water concentration is less. You all know that due to osmosis the water molecules will move from higher concentration to lower concentration. So you can see in this figure that yellow arrow, what it is showing? It is showing that water is transported from outside into the cell. As a result, the RBC swells up, and eventually what happens? When the pressure increases inside the cell, when the osmotic pressure increases inside it, the cell bursts. So this type of this is known as plasmolysis. This is not known as plasmolysis. As a result, what happens? The cell eventually bursts because the osmotic pressure inside the cells increases, so it cannot bear up and it bursts. Okay, the second type of solution is isotonic solution. Okay, in isotonic solution the outside cell and the inside of the cell is having equal concentration. So if the medium which is surrounding the cell has exactly the same water concentration as it is inside the cell, then there will be no movement of water molecules, because on outside of the cell as well as inside of the cell the water concentration is same. So such type of solution is known as isotonic solution, and the cell shape will not change, it will remain same. So you can see in the slide we have taken RBC and we have kept it in isotonic solution. So the amount of water transported into the cell is equal to the amount of water coming inside the cell. The yellow arrow is showing the amount of water going inside the cell and green arrow is showing the amount of water coming outside the cell. So since both the concentrations of the water inside and outside of the cell is equal, so we can say that the cell size remains same. It is not going under any changes. So this type of solution is known as isotonic solution. And the last type of solution is known as hypertonic solution. Hyper means more. Now whose concentration will be more? It is always the concentration of the solutes. So since the concentration of the solutes in the solution is more, so it is obvious that the solution will have less concentration of water. So when the RBCs, you can see here in hypertonic solution, so when the RBCs are kept in a hypertonic solution which is having less water and more of salts, what happens inside the RBCs? The concentration of water is more. Okay, fluid concentration is more, and outside it, since it is less, so automatically what happens? The water from the red blood cells start coming outside. You can see in the diagram, this green color arrows are showing the movement of the water molecules from the cell outside to the outside environment. As a result, what happens? The cells shrink, they decrease in their size, and this process is known as plasmolysis. So the process by which the cell shrinks when they are placed in a hypertonic solution, such type of process or shrinkage of the cells is known as plasmolysis. So when you are placing a cell in a hypertonic solution, it causes shrinking of the cell, and that process is known as plasmolysis. So we have seen that there are three types of solutions, hypo, isotonic and hypertonic solution, and how it regulates the shape of and size of the cells. Okay, now we will see that since we have seen that how the molecules, whether it is gaseous molecules or it is water molecules, it is passing inside the cell with the help of cell membrane or plasma membrane. Okay, now we will see that how the food materials, because you all know that cells are living cells, so they need food to survive, they need other organisms, or if any foreign particles enter into our body, they need to engulf those particles. Okay, so to perform those performance or to engulf those substances, it undergoes another kind of process which is known as endocytosis. Okay, so we will see what is endocytosis. Okay, so when any kind of substance enters into the cell, then it is known as endo means in, so inside the cell. And when any kind of substance is going outside the cell, then it is known as exocytosis. Exo means out. So first we will discuss what is endocytosis. Okay, you can see here in the slide, the endocytosis is nothing but it is a process, it is the flexibility of the cell membrane which enables the cell, means which helps the cell to engulf the food and other materials from its external environment. When the cell needs food, it will be nutrition, it will be any solid substance, or when suppose any pathogen has entered into the body or any microorganism, then it will be other materials. So the ability of the cell membrane or the ability of the cell which is being enabled by whom? Cell membrane, to engulf the food material or any other materials from its external environment is known as endocytosis. Okay, we will if you will go further in classes, you will come to know that amoeba is a single cell animal, isn't it? It is a unicellular organism, so it undergoes all the process like taking up of food, water, everything. It acquires food with the help of endocytosis, because it needs to take the food from the outside environment into its cell. So amoeba acquires its food through this process. Okay, now endocytosis are of two types. The first one is known as phagocytosis, which means that cell takes in solid food material through the plasma membrane. So if any kind of solid thing is passing through the plasma membrane inside the cell, then it is said to be phagocytosis. Okay, on the other hand, the other kind of endocytosis is pinocytosis. So if the cell takes fluid kind of matter through plasma membrane, then such type of process or phenomenon is said to be pinocytosis. So endocytosis is categorized into two, phagocytosis and pinocytosis, based on the type of the material it is taking in. If it is food, then it is phagocytosis, solid, and if it is liquid or fluid, then it is said to be pinocytosis. Okay, so this is the this in the slide you can see the diagram of endocytosis. Okay, you can see the cell structure, cell is being shown, and cell, you know that every cell is having an outer covering which is known as plasma membrane. In the first diagram you can see that yellow color, lining is cell membrane. Okay, in the second diagram you can see that green color outer covering is nothing but it is plasma membrane, or you can call it as cell membrane. Okay, you can see the extracellular fluid in which the cell is present. Okay, so in the second diagram you can see that endocytosis is taking place in the animal cells. Now endocytosis may also take place along with the receptor mediated receptors. There are certain big molecules like proteins. So proteins cannot enter into the cell. They need receptors which will identify those proteins. They will attach themselves to the proteins and then they will push them inside the cell. So we can see in the second diagram that first one that said that plasma membrane undergoes invagination. It has having receptors, that red colors bilobed structures are receptors which are holding those particles and they are shifting it inside the cell membrane. Okay, and those are being packed in the form of vesicles. Okay, and those black color are the coat proteins which are present in the plasma membrane. You all have seen yesterday that plasma membrane also have proteins on its surface. Okay, then in the second type, pinocytosis, you can see those fluid molecules. Those green color are the fluid molecules. Okay, so those fluid molecules are being entering into the cell and then they are also being packed into the vesicles. Okay, and the third type is phagocytosis. As I have already said you that it is the food material. If it is solid food material, then they will engulf it and they will pack it in the form of a vesicle, and that vesicle is known as food vacuole, which is generally taking place in case of amoeba. In amoeba you will find so many food vacuoles. Okay, and the process is nothing but endocytosis. Okay, phagocytosis is also used for the killing of the microorganisms. It is the special features of cells. Okay, you can see in the second slide endocytosis. First one is phagocytosis, how the solid particle is being entering through the plasma membrane and it is forming phagosome. The other name of food vacuole is phagosome. It is generally found in neutrophils. They have phagocytic functions. Okay, next is pinocytosis. It's showing the entering of the fluid substances which enters into the cytoplasm and it takes the form of a vesicle. Okay, and the third type is receptor mediated. I have told you that some larger proteins, macromolecules like proteins, they cannot pass the plasma membrane. Okay, so in order to pass through the plasma membrane, they need special receptors. They need to be identified by receptors which are present on the surface of the plasma membrane. So as soon as they will be identified by the receptors, they will be pushed inside the cell membrane, and then you can see that there are coated vesicles formed which is having a coat protein on their body wall. So these are the three types of endocytosis which are being seen in the cell of any animal. Okay, and the last one is exocytosis. Now it is the process of exudation. Exudation means elimination of the secretions or the waste materials out of the cell through plasma membrane. Okay, it is the opposite of endocytosis. You all know that endocytosis, endo means in, endo means to take in. So since exo means to throw out of the cell, so exocytosis is just the opposite of the endocytosis. Okay, and it is the process of elimination of secretions, because cells sometimes secrete useful substances like interferons. When the virus infected cells are, when any cell is inside our body gets infected by a virus, it starts secreting interferons. Okay, interferons are a group of proteins that helps the other cells to inform that the cell is being undergone virus attack, so that they can protect themselves. So if it is any kind of secretion, or if they need to excrete any kind of harmful substance from the cell like gaseous products and all, then they undergoes exocytosis. Okay, this is all about the types of solutions and how the food materials or the fluid materials enter inside the cell, or it's being excreted outside the cell. Then I have told you that in plant cell, in addition to the cell membrane, another one outermost covering is present, which is known as cell wall. So it is a peculiar characteristic of all the plant cells. It is not present in animal cell. It is only and only restricted to the plant cells. Okay, so the non-living, it is the non-living rigid structure that forms the outer covering from the plasma membrane of plants and fungi. So in animals you will see only and only plasma membrane, but in case of plants and fungi you will see an extra outermost covering which is known as cell wall. So cell wall is actually dead, it lies outside the plasma membrane and it is mainly composed of a cellulose. Okay, now what are the functions? So there are so many functions of the cell wall, such as it not only gives shape to the cell but it also protects the cell from the mechanical damage and infection. Since it is the extra covering which is given to the cell, so it helps the cell, it protects the cell from the external damage and injury, okay, and infection as well. But at the same time it also helps in cell to cell interaction, from one cell to another cell it helps in the interaction, and it also acts as a barrier to the undesirable macromolecules. You must have seen that in plants one cell is attached to the another cell with the help of a connection which is known as plasmodesmata. So we can say that cell wall helps in the cell to cell interaction. It also acts as a barrier to the macromolecules, the molecules which are very large in structure shape. They cannot pass through the cell wall, so they get obstructed by the cell wall. So these are the certain functions, like it gives shape to the cell, as I've already told you. It gives rigidity, means strength, hardness to the cell. It gives mechanical strength to the cell. It also supports the internal structure of the cell because it acts like an additional protection. It also protects the cell from osmotic bursting, isn't it? In case of animal cell you have seen that RBC swells and it bursts, but in case of plant it generally cannot get burst because it is having additional cell wall. Then, and the last part is it helps in the expansion of cells. Okay, this is how this last diagram is showing. This is how the plasma membrane undergoes invagination, and it helps in the exocytosis. See, there is all the molecules or which is need to be excreted or secreted. It is present in the form of vesicles. Okay, these vesicles are being secreted by whom? Golgi apparatus. In next class we will see that Golgi apparatus is a cell organelle which helps in the packaging of materials. Whatever materials need to be secreted or excreted, they are packed in vehicles, like how we whatever food we eat, whatever food we take from the market, we have in separate packets. Just like that, inside the cell every material is packed inside the vesicles. Okay, now these vesicles gets attached with the plasma membrane. It fuses with the plasma membrane and it liberates those materials outside the cell in the extracellular fluid. This is the process of exocytosis. Okay, and lastly we will see plasmolysis. As I've already told you that when a cell is placed inside a hypertonic solution, hyper means more of salt and less of water. Okay, then what will happen? The cell will start releasing its water from inside of it to the outside environment. Okay, as a result what happens? The cell shrinks in size and that process is known as plasmolysis. Okay, see in the diagram we have taken some bean seeds. Okay, in those bean seeds if you add 20% sucrose, that means it is become hypertonic, isn't it? Hyper means more of salt and less of water. As a result what happens? After 2 minutes you can see what you can see that the that it has undergo shrinkage because outside the concentration of salt is more and water is less. So as a result whatever water is present inside those beans, it will start going outside the cell. Okay, so that it will shrink and it undergoes plasmolysis. On the other hand, you can see that when you add distilled water in the surrounding environment, then what happens? It start entering into the cell because it becomes hypotonic. That means more of water and less of salts. So the beans again swells up. Okay, and the last one is protoplasm. All the living components of a cell, that means you know that some are non-living. Suppose cell wall is non-living, but you see cell membrane is living, nucleus is living. So all the living components of a cell is called protoplasm. It is colorless, it is translucent, it is jelly like viscous semi-fluid substance. Okay, it generally contains a centrally located nucleus and cytoplasm. So you can say that nucleus and cytoplasm together forms the protoplasm of a cell. Okay, This is how the plasmolysis is taking place. It is the figure showing when the plasmolysis takes place, the the cell shrinks, and the cell membrane moves away from the cell wall. Okay, this is plasmolysis. So this is all about today's.